Please note information Dr. Morton put up there about lab work. Was there anything else you needed to say?
Except to emphasize that those of you who had lab on Tuesday, you've got blue and white colonies to pick. They're pretty big. I don't want them to go another day, so you've got to come in today to take care of that. Those of you who had lab yesterday, many of you have good-sized colonies that you can pick today, but if not, tomorrow by noon, please. And those of you who have lab today will have to wait until Monday to pick the colonies.
But it literally just takes a minute to grab a colony and put it into a tube of broth. Little heads up, the last day of last week of class during lab, we're going to be doing the course evaluations, and those are all done online right now. So we'll be done in the lab. We have, 7 computers in the lab? So that isn't enough for everybody. So if you have a device you can bring that you could log on to and do your evaluation, that would be appreciated. If not, we'll just have to do it in kind of groups. So we'll remind you prior to the lab. But
well, for Monday and Tuesday especially, Wednesday
and Thursday we've got enough. Wednesday and Thursday we should have enough computers, right?
Well, there's 8 on Wednesday and 6 on Thursday. So
Okay.
Close.
Your quiz Monday will be over a very defined set of material. It's just going to be over the operon, slack, and trip. So that page is 293 through 297 in your textbook. So any questions? Oh, did the slides come through that I sent you?
Yes.
Okay. It was a pretty big file, and I wasn't sure if that was going to be a problem. There are not significant differences between the ones I sent you and the ones that were posted, but I reordered a few things and made a few clarifications, so those are the best ones to use. Oh, I thought that was a fire alarm. So we spent a fair amount of time talking about the lac and the trip operon and the way that bacteria can respond to changes in their environment through changes in gene expression, turning on and turning off genes as needed. And that's really an important aspect of their ability to survive and adapt to environmental changes. When we start talking about regulation of gene expression in eukaryotes, it gets a lot more complicated because the eukaryotic genome is more complicated. For example, instead of having just a single circular chromosome like bacteria do, humans have 23 homologous pairs of chromosomes. The chromosomes themselves are much more sophisticated and complex than our bacterial chromosomes. And then you've also got the fact that in eukaryotic cells, you've got the chromosomes housed within a nucleus. There's no nucleus in a prokaryotic cell. We've got those different methods of processing RNA that happen in eukaryotes. Those don't happen in prokaryotes. So what we're going to do is we're going to give you just kind of an overview of some of the ways, additionally, besides operon-like regulation, that your cells of your body can regulate gene expression. And it's really important that they are able to do so. Here's one reason why. Pretty much every cell in your body, there are a few exceptions, particularly the gametes, are genetically identical to one another. In each cell of your body, whether it be a liver cell or a skin cell, are all the chromosomes and all the genetic information needed to make all cell types, not just skin cells and liver cells. The information to make a complete, intact organism. So how do cells become different from one another? How do we develop these muscle cells and brain cells and things? We have to have differential gene expression in those cells. We have to have precise, orchestrated turning on and turning off genes at critical times, particularly as the organism is developing from that single cell zygote that we all start as, through our embryonic stages, through our fetal stages, and into childhood and adulthood. In a given cell, and this is a pretty gross just estimation, only about 20% of your total genes are active at any one given time. The rest are turned off. Why? Because they code for other cell types. What's kind of scary is that if the gene expression changes don't occur in the proper sequence at the proper times, then that can lead to some pretty debilitating conditions, including birth defects and the development of cancer. And this is one we're going to follow up on a little bit later on, is how errors in gene expression can result in the development of cancer. So, what are the opportunities that exist in eukaryotic cells for regulating gene expression? Well, they have gene expression regulated at the operon level. But in addition, here are some other opportunities that are present for regulation of gene expression in eukaryotes that aren't present in prokaryotes. We're going to kind of go through these one at a time, but I wanted you to have this kind of list so you can see kind of vast possibilities for regulating gene expression that occurs within our cells. And then this one down here, the involvement of non-coding RNAs in controlling gene expression, is a relatively new understanding about how gene expression works and the importance that RNA plays in regulating expression of DNA into RNA. So it gets a little bit complicated. So we're going to scratch the surface but not get into a huge amount of detail about this. So basically what we're going to do is we're going to work our way through the list and talk about each one of these in order. So, regulation of chromatin structure. Do you remember that term chromatin and what it refers to from last semester? Where did you encounter the term chromatin? Hint. Think mitosis and meiosis. Hint. Think interphase. What is interphase? What's happening to the chromosomes during interphase? Pardon me? Not yet. Remember, interphase is the part of the cell cycle that's sometimes called the resting phase of the cell. And it's not resting, though, because a lot of things are happening in the interphase nucleus. One of the things that's happening, were you going to say something, Ricky?
Yeah, the DNA is coiling up. The chromosomes are coiling.
They will start to coil at the end of interphase. But then when we get into the mitotic phase, the M phase of the cell cycle, that's when the chromosomes start coiling up, migrating to the cell equator, and splitting. Okay? And when that happens, the chromosomes change shape. They condense, they shorten and thicken. In the chromatin state, the DNA is kind of all twisted up. It's kind of like this thread-like conglomeration of DNA molecules. During interphase, a lot of those genes are being expressed. They're being transcribed and translated. That typically stops once we get into the M phase, and we stop temporarily transcribing those particular genes. We'll see why in just a second. So, chromatin refers to kind of the state of DNA as it's associated with various proteins in the interphase nucleus of the cell. Well, if you break down the structure of chromatin in a little bit more detail, one of the things we'll see is that there's kind of a repeating unit of chromosome, kind of a basic organizational unit that makes up this material called chromatin. And this basic organizational unit is called the nucleosome. And we'll show you what a nucleosome looks like in just a second. One of the things that helps to maintain nucleosome structure are some proteins called histone proteins. So here's some key words right here. Chromatin is kind of made up of repeating units called nucleosomes, and histone proteins are important in the structure of these particular nucleosomes. How does this relate to regulation of gene expression? Well, as it turns out, the way chromatin is folded up is going to have an effect on how the genes that are part of that DNA are expressed. In some states of chromatin, expression of genes is almost non-existent. When chromatin is in another state or shape, then genes are being transcribed fairly frequently and fairly regularly. So, what's going on here? Okay, most people kind of assume that a chromosome is just a DNA double helix. So when you start thinking about those chromosomes being lined up on the equator of the cell during mitosis, you're probably more or less kind of visualizing, well, those chromosomes are just DNA double helix. And they're not. They're much more complicated than that. Because chromosomes are not only made out of DNA, but there are several proteins associated with chromosomes as well. And the most important kind of protein from our purposes right now are these proteins called histones. And what do? They can actually bind to the DNA. And clusters of histones can come together and attach to one another, forming kind of a glob of proteins. And then the DNA coils around those particular histone proteins. This coil of histone proteins and DNA is what we call a nucleosome. And so this represents chromatin in a pretty relaxed state where we've got stretches of DNA and then nucleosomes and then stretches of DNA and nucleosomes. Here's an electron micrograph of chromatin in this particular state, this kind of relaxed state, and you can see these repeating nucleosomes represented by the histone proteins and in the intervening DNA. Nucleosomes are also called beads on a string because this really looks like kind of a string with beads on it. Well, in reality, DNA folds up even more than this. These stretches of DNA tend to become coiled up into masses of DNA and protein. These masses of DNA and protein tend to form these loop structures, and these loops all aggregate with one another. And that really is what a chromosome is consisting of. So DNA is highly folded when it's in its chromosomal state. The structure of DNA is pretty relaxed when it's in its chromatin state, when it's that kind of series of thread-like twisted DNA molecules. Once we get into prophase, then the chromosomes start to condense, they start to further fold up, and then we can see actual visible chromosomes kind of emerge from that chromatin mass. So what's this all got to do with regulation of gene expression? Well, basically chromatin can exist in two general states or forms. And one is loosely packed DNA, and one is tightly packed or all twisted up and coiled DNA. Okay? A metaphase chromosome would represent very tightly packed DNA. Loosely packed DNA is called euchromatin. So here's some terminology you're going to have to remember for exam. Tightly packed and twisted up DNA is called heterochromatin. And what's the relationship between gene expression and these states of DNA? Loosely packed DNA, or euchromatin, is pretty easily transcribed into messages. When the DNA becomes all coiled up and twisted up, it's much harder to transcribe it. So actively transcribed, hardly ever transcribed. Well, if you look back at this illustration right here, it kind of makes sense. Okay? So this would represent euchromatin. Very relaxed, not all twisted up DNA. This would represent more highly compacted DNA. What has to happen in order for transcription to occur? An RNA polymerase molecule has got to recognize and gain access to a promoter associated with a particular gene. Where's it going to be more likely that it's going to be able to gain easy access when the DNA is all twisted and coiled up, or whether it's in this loose, relaxed state? Here, okay? So in this particular euchromatin state, the DNA is pretty easily transcribed. In this more coiled up, twisted up heterochromatin state, it's not going to be transcribed hardly at all. And especially if you get to this state right here, where the heterochromatin folds back on itself and finally forms these complex, highly folded structures called chromosomes. So there's really no transcription occurring during mitosis. The chromosomes are just too wadded up for those RNA polymerase enzymes to gain access and transcribe those genes. So this represents an opportunity for cells to regulate gene expression if they can somehow regulate whether their DNA is in this loose state or in this highly packed state. Make sense? Well, there are methods that the cell can use to regulate the state of chromatin. And it involves, not surprisingly, some enzymes. There are enzymes that can actually chemically modify chromosomes. One of the chemical modifications can occur to the histone proteins. Remember, those are the proteins that interact with DNA and cause those nucleosomes to form? Another set of enzymes that we'll briefly talk about actually interact with the DNA part of the chromosome. But these enzymes can chemically modify either the histones or the DNA itself and influence whether those DNA molecules and proteins are all globbed up, heterochromatin, or are relaxed, euchromatin, and in that state they can be pretty easily transcribed. This state, they can't. The chemical modifications of the histones include two processes, acetylation and methylation. And we're going to talk briefly about acetylation, just mention it. Chemical modifications of DNA involve methylation. So there are enzymes that can grab these functional groups called acetyl groups and stick them onto histone proteins. That's going to affect the shape of the chromatin. There are enzymes that can grab methyl groups and stick them onto DNA, and that's going to affect the shape of that particular genetic material. So here's what we're looking at. So in histone acetylation, these acetyl groups, you don't have to remember this, you don't have to memorize the structure, are attached to the positively charged lysines in histone tails. So these functional groups are negatively charged, and they're attracted to the positive portion of a histone molecule. When this happens, this loosens chromatin structure, and that facilitates transcription. So histone acetylation loosens DNA structure and facilitates transcription. So here's what it looks like. So here's a nucleosome. Okay, here's a bunch of histone proteins with some DNA wrapped around it, those beads on a string. Okay? These tails of the histone proteins contain a lot of positively charged amino acids. And these acetyl groups, which are negatively charged, are attracted to those positive charges on those amino acids. So if we take some DNA and some enzymes acetylate those histones, it causes the structure of the chromatin to change from the compact state into the relaxed state. Heterochromatin into euchromatin. Not being transcribed, now the enzymes can gain access to it and be transcribed. So here are some enzymes that can regulate gene expression by interacting with proteins that are themselves interacting with DNA. Told you it gets a little bit complicated. Okay, another set of enzymes that don't react with the proteins, histones, but react with the DNA itself. in DNA methylation, a methyl group can actually be physically attached to some of the bases of DNA. The most common one is cytosine. And when we methylate DNA, it tends to cause the DNA to become more in that heterochromatin state and to therefore be less transcriptionally active. So DNA methylation tends to prevent transcription, to tighten up the structure of DNA. Sometimes cells will use DNA methylation to almost permanently glom up genetic material so it's never transcribed. And the chromosomes become pretty much permanently inactive because their DNA is highly methylated. Have you ever heard of bar bodies before? Did you guys talk about bar bodies last semester?
I did.
You did. Those of you that had Dr. Morton should be able to answer the class, right? Good question? Who has bar bodies in this class? Does Ricky have them? Does Monica have them? What's happening? What is the genotype of a female in regard to XY chromosomes? What is the genotype of males in regard to XY chromosomes? Males are what? XY. What are females? XX. So females have twice the dosage of X-linked genes as males, right? That could potentially be problematic. And one of the things that happens during cell division and development is that one of the two X chromosomes becomes highly methylated and becomes basically condensed permanently into this inactive X chromosome. That's called a bar body. And so the genes on that chromosome aren't being transcribed. And you can actually see that if you do proper staining of cells. And here is a female cell with this permanently, all the DNA here is in the chromatin state, so pretty transcriptionally active, pretty loose structure, okay? But there's this permanently condensed X chromosome, the bar body, that's basically just been kind of taken out of the game. Why? Because the DNA has become highly methylated. Well, what's kind of interesting is that once genes become methylated, they typically maintain that methylation pattern through successive cell divisions. So daughter cells inherit the methylation pattern from the parent cell. And that has some interesting implications. These modifications to chromatin, this methylation, isn't changing the sequence of the bases in the DNA. It's not changing the order of the A's, G's, T's, and C's. But because it's affecting the shape of the chromosome, and therefore whether it can be transcribed or not, this methylation is affecting gene expression. And those patterns can be passed on to future generations. We call that particular type of inheritance epigenetic inheritance. And epigenetics is becoming a really hot topic right now. Epigenetics refers to the inheritance of traits transmitted by mechanisms that don't involve changing the nucleotide sequence. Okay, it's not the same as mutation. So, for example, identical twins have the same DNA. Why does one twin potentially develop a disease that the other one doesn't? Like a genetic heart-based disease like schizophrenia. They have the same genes. Because they're being expressed differently, because they have different DNA methylation patterns potentially. This whole idea of epigenetics is really kind of interesting because one of the implications of this kind of inheritance is sometimes environmental factors can influence expression of genes in individuals. And if that involves alteration of chromatin structure, those patterns can be inherited. So actually the environment can cause changes in gene expression which are heritable. That's something we didn't used to think could happen. In fact, did you talk about Lamarck when you talked about genetics? Okay. What's also interesting from our point of view later on is that alteration in normal DNA methylation patterns is sometimes seen in cancer cells. These changes can cause inappropriate gene expression, which can lead to the development of the state of cancer. I just wanted to throw that term out there for you so you've heard of it, because this is something that's going to be important and relevant. You will dig into it much deeper if you go on and take Dr. Morton McComb's genetics class. Okay, so one opportunity for regulation of gene expression is regulating the shape of chromatin, making it compact or making it loose, by either acetylating histones, which loosen and promote transcription, or by methylating DNA, which tightens up DNA structure, which tends to shut off transcription. We can also regulate transcription at the stage of initiation of transcription. Now this one gets a little bit hairy, and I'm trying to keep it as simple as possible while still giving you the basic idea of how this particular might work. I'll show you a diagram in just a second that'll flesh out some of these things. But there are some control elements that are going to be involved, particularly these regions in the DNA called enhancers, which can have a very important influence on whether genes are transcribed or whether they're not transcribed. So most eukaryotic genes have these things called control elements that help to regulate transcription. And they're segments of DNA that we call non-coding DNA. Now, that's an important term to kind of wrap your head around. When we refer to non-coding DNA, those are stretches of nucleotides that will be transcribed into RNA. But the RNA isn't messenger RNA. It's some other kind of RNA. Some examples of non-coding DNA are ribosomal RNA genes, transfer RNA genes. Those don't really have much to do with regulation of gene expression. We're going to talk about a non-coding DNA later that codes for a type of RNA called microRNA, and that is going to have some relevance to regulation of gene expression. But these don't code for polypeptides. That's what we mean when we say non-coding. It doesn't code for polypeptide chain structure. There are two types of control elements, non-coding DNA regions, that really are important in regulating gene expression. And they're called proximal control elements and distal control elements. Anybody in here, some of our exercise science AT people had A&P, right? What do proximal and distal mean? Close and far away. Okay, proximal means near or adjacent. Distal means far away. So, here's what things might look like. And I know this looks a little bit daunting and confusing. But basically, here's a gene. What are exons? Or more importantly, what are introns? Remember, introns are the non-coding information that splits the coding segments of a gene. So these are the split genes. Okay, so here's a gene with 2 introns and 3 exons. Here's a promoter. What's the relevance of the promoter to transcription? That's where the RNA polymerase attaches, remember? Okay. But then there are going to be some control elements that are going to influence the ability of RNA polymerase to attach to and recognize that promoter. There are some that are nearby and there are some that are far away. So proximal control elements and distal control elements. If the right proximal and distal control elements interact properly, then RNA polymerase can attach to this promoter and transcribe this gene. We make a message. That message is then processed. The introns are cut out. Remember, the cap is added. The tail is added. And then we have a message which can be translated into a protein. That gene has been expressed. So really, initiation of this process of transcription is a real good point at which we could regulate gene expression. If things prevent RNA polymerase from attaching to this promoter region, the gene won't get transcribed. If something facilitates RNA polymerase from attaching to that promoter, then the gene is going to get transcribed very readily. Well, these control elements, other sequences of DNA, are going to be important in determining whether transcription occurs or not occurs. So, if we're going to transcribe genes, the eukaryotic RNA polymerase requires some assistance. It requires some proteins. And those proteins are called general transcription factors. Now, I mentioned that before when we talked about transcription. I said, in order for RNA polymerase to work, it needs the help of some proteins, transcription factors. Those transcription factors are going to be interacting with one of these proximal control elements, and that's going to help to facilitate the RNA polymerase attaching and therefore transcription occurring. If these general transcription factors don't get involved, transcription's not going to happen. The polymerase enzyme needs some help from those transcription factors. Now, just because you have these general transcription factors involved doesn't mean the rate of the transcription is going to be very fast. Usually, to get a decent rate of transcription, you need some specific transcription factors also involved. And these specific transcription factors are called activators. Does that sound familiar? Yeah, that sounds like the positive gene regulation we talked about in the lac operant. Remember we said that in the presence of lactose, that we get a slow rate of transcription of that lac operant? But you'll get a faster rate of transcription if that catabolite activator protein is involved. That'll increase the rate of transcription. That's an activator protein. Well, these activators, these specific transcription factors, interact with those distal control elements, not the proximal ones. Okay? So the general factors interact with the proximal control elements and facilitate transcription. The activators interact with these distal control elements and really speed up the rate of transcription. In order to get good gene expression, typically both types of elements have to be involved. So, let's concentrate on these activators a little bit. These things that really speed up the rate of transcription. What is an activator? It's a protein, also a specific transcription factor. and it's binding to a distal control element, and it's going to enhance the rate of transcription. These proteins have two parts. A fancy word for part is domain. One of the parts of this activator binds to DNA. The other part interacts with other proteins that are going to help to activate transcription. Here's kind of what a activator molecule might look like. So this represents a polypeptide chain. Here's one domain that interacts with a section of DNA, one of the control elements. And then here's another domain or part of that protein, and this is going to interact with other proteins. Told you it's a little complicated. So, let's see what this might look like here. So, here's our promoter. RNA polymerase has to be able to attach to this promoter in order to transcribe this particular gene. It needs some help. It needs some general transcription factors. To recruit these and get them to become involved in helping RNA polymerase, we also typically need some activators. Okay? Those are the molecules I just showed you. The proteins that have two domains. One allows them to bind to DNA, this distal control element, and the other domain allows them to interact with other proteins. So what if I've got the proper activator? That binds to the distal control element. And then that brings in or recruits other proteins. One of the proteins that it recruits is this interesting protein called DNA bending protein. And this binds to the DNA and causes it to fold back on itself. What does that do? This distal enhancer, which was far away from the promoter initially, is now in close proximity to the promoter. It's brought back into close association with it. The protein binding domain of these particular enhancers now interact with some other proteins. They're called mediator proteins. And when they bind, they help to then recruit these general transcription factors. And now the RNA polymerase can come in and transcribe the gene. So look at all the different elements that have to be involved to get a good rate of transcription. Mediator proteins, bending proteins, general transcription factors, activators. And if the cell can somehow control the availability of any one of these things, it has ways in which it can control the expression of genes. One of the things about studying the molecular basis of gene expression is the more you start learning about it, the more questions you have, and the more complicated it really becomes. But this just gives you some kind of an idea of all the different opportunities there are now for regulating gene expression by the availability of certain key elements like activators or mediator proteins or general transcription factors. So, an application. A very practical question. Let's take a look at two types of cells. One is a liver cell. And the other is a cell in the lens of your eye. That clear lens of your eye. Liver cells produce a lot of a protein called albumin. That's one of the things that characterizes liver cells, is cranking out a bunch of albumin. Lens cells of the eye produce this clear protein called crystalline protein. Why does the liver cell tend to produce this, but not this? Why does the lens cell tend to produce this, but not this? Don't they both have the same genes? They do. So why are some genes being preferentially expressed in liver cells and some in lens cells? One of the things that can help to explain that is the presence of activators in different cells, different activator molecules in different cells. All the activators needed for high-level expression of albumin genes are present in the liver cells, but those same activators aren't present in the lens cells. Vice versa. Activators for expression of crystalline gene are present in the lens cells, but not the liver cells. During development, one of the things that happens is that these activator molecules get kind of apportioned or separated out into different cells during the process of cell division. So it's not uncommon for different cells to have different proteins. So here's two genes. Both have these distal control elements, enhancers. Here's the albumin gene, here's the crystalline protein gene. Both types of cells have both of these genes. But in the liver cell, this gene is being expressed, this gene isn't. In the lens cell, this gene is being expressed, and this one isn't. Why? Because the lens cells don't have the same proteins as the liver cells do. They don't have the same activators. Here, if you have the proper activators, they'll bind to this distal control element. That will recruit the DNA bending protein, the mediator proteins, and the transcription factors, and that gene will be expressed.
This is like a genetic experiment type deal. So if you could find the right activators and proteins needed and somehow induce them into a liver cell from, could you make a liver cell produce an eye lens almost?
Mm-hmm. Yes, you can.
Is there any practical application?
Any practical application of having liver cells produce crystalline protein?
Or just like
Not that I can think of. Larry, you looked like you were going to say something.
The whole science of using and creating stem cells is to reprogram. For example, you can take a fat cell and reprogram the DNA so now it can become a liver cell or a lens cell. So by being able to go into the DNA of the cell and change its methylation patterns and then reinitiate the process of differential gene expression is how stem cells, whether they be fat cell derived or embryonic stem cells, are used to produce new types of cells for therapeutic use.
Yeah, it's interesting too because let's finish the scenario and then we'll talk about something else for a second. So in the lens cell, again they've got the same two genes, but they've got different activators, different proteins, and they're not the activators present that cause this gene to be expressed, but there are the appropriate activators to cause this particular gene to be expressed. One of the things that's really always a question is why do these different cells have different activators? Why do they have different proteins? And part of that has to do with position of the cell in tissue types in different parts of the body. And one of the things you can do interestingly in embryology experiments is you can take cells out of one part of the body and transplant them into another location, and that seems to influence which proteins they have. And then all of a sudden in this new environment they can produce new activators and they can start differentiating into new tissue types. What was the example you gave me the other day?
Oh, why do birds not have teeth? If you take the tissue that covers the gum of chickens and transplant it onto a similar spot in a developing mouse embryo, the chicken epithelial cells will make teeth because they're getting specific signals from the underlying mouse tissue.
Yeah.
And those signals involve the generation of those activators or transcription factors. So chickens can make teeth, just don't get the right signals.
So, it raises all kinds of interesting questions if you think about it, because these proteins all influence gene expression. But remember, proteins are the product of gene expression themselves. And so molecules regulate gene expression, but they themselves are under regulation of gene expression by other mechanisms. And so that's why it gets so complicated and so hard to wrap your head around all of the complex interactions that are taking place to make this possible. But this is just a very basic kind of example that gives you an idea of how two cells with the same DNA can express different genes based upon the presence of certain key protein molecules. And it all has to do with regulation at the transcriptional level of initiation. Well, we have some other opportunities to regulate gene expression, and that'll be something we can pick up on tomorrow. And some of these refer to things we've already talked about, RNA processing, for example.